Okay, let's get connected. First of all, thank you very much for the invitation and giving me the opportunity to speak here in front of this audience. I'm going to switch subject a little bit, so away from the therapeutics now to a bit of different theme. I'm going to talk about tumor genome-based treatment decision support. But before I actually get there, I change subject again. I talk about IT. The picture you see here, that's actually a light sculpture that you can find in the Nixdorf Computer Museum. And what it actually symbolizes is the Moore's Law. We all know the Moore's Law from IT. So Moore's Law tells us that processing power of CPUs doubles every 14 to 24 months. So it's a symbol of growth, it's a symbol of exponential growth that actually drives an industry. And if I now look at the analogies to the field that we are all in, to the biomedical world, I can observe something really similar these days. With the advent of all these high throughput technologies and specifically sequencing, next generation sequencing, and now next generation sequencing, we are in a pretty similar situation. Actually, in our science, we outbeat the Moore's Law. So we have made such tremendous progress coming down from 3 billion for the first genome now to a price tag and a speed that actually makes sequencing affordable and available. And it's not a surprise that sequencing nowadays finds its way into the diagnostic field, and we are just at the beginning of that. This application of the Moore's Law, I mean, is actually complemented. You can also apply the Moore's Law to the biomedical field in general. There has never been a scientific field that generates so much progress as the biomedical sciences these days. We have an absolutely exponential growth in new findings. And I think I need to connect power. And what we actually need to do is marry these both fields. So high throughput data generation doesn't get us any further with respect to clinical insights if we don't combine it with a biomedical knowledge and keep it up with the current biomedical knowledge that we are generating. So really the challenge is merging those two worlds together. What happens if we don't do that is exactly this. So this is actually not just a cartoon. This is something that happens in reality. And I've had many collaborators at MD Anderson in Houston, Texas, who have told me they've had patients coming to them. They brought their hard disk. They said, here is my tumor genome. What can you do for me? So actually, it's pretty hard for a physician to tell from all these GA/T&Cs what they should do with a patient. So they need a reference system. They need to isolate from this massive amount of data the information points that really make a difference and that are clinically meaningful. And that's actually what we are trying to do as a company. So we originate from the bioinformatics field, and what bioinformaticians really love doing is data integration. So that's a perfect field for us. If we want to identify signals from the cancer genome that are clinically meaningful, we need to understand and we need to model the molecular mechanism of the disease. We need to understand what are tumor drivers, what are biomarkers, what different kinds of biomarkers are there. We need to understand structure-function relationships in proteins that are affected by mutations. On the other hand, if we want to give treatment recommendations, we need to understand the molecular mechanism of drugs. We need to understand that in detail. So it's not good enough just to know a drug interacts with a target and inhibits it. We also need to know about off-target effects. We need to know about metabolizing enzymes. We need to know about transporters, because all this information is also important, because we're not only looking at treatment efficacy, we're also looking at safety. And safety is an important field, especially in oncology when we're dealing with these toxic drugs. So we're integrating all this data, we are matching that to biological concepts that are actually meaningful for treatment decision making. And what we're coming up with, and this is at the top of the pyramid, and actually that symbolizes our intent. Our intent is really to reduce the search space, to make the oncologist focus on what is clinically meaningful and significant from all the data that is contained in that massive amount of sequencing data. And what we want to derive from this is information about what are suitable targeted therapies, what are combination therapies, even chemotherapies, or if we don't have any approved drugs that show up, are there any clinical trials that fit to the patient's genomic profile? Is there any treatment resistance? And also actually risk classification and adverse events are important. So how does that work in practice before I get to the detail? So we have implemented our treatment map in a, I mean, it looks like a fairly complex process, but it's actually really simple. So we have the patient and the physician, and the process actually for genome sequencing and then the clinical interpretation of that is always accompanied by a trained oncologist, or in the US, by a molecular pathologist. So we have the sample logistics, tumor sample, and if we do exome sequencing paired, also blood sample for solid tumors, they are subjected to sequencing. And actually, in Germany, sequencing is done by one of our partners, GATC in Constance. In the US, we are operating for regulatory reasons our own NGS sequencing lab, which by now is CLIA certified. Then comes our special interest, which is the interpretation, the analysis and clinical interpretation to optimize efficacy and safety of treatments. And at the end of this chain, actually there is what we call treatment map report that's actually signed off by a trained oncologist, or in the US, a molecular pathologist, and then actually given back to the treating oncologist. So let's look at what we do with the data. The first steps are pretty simple. We get the raw sequencing data. We do nothing different than everybody else in the world is doing. We try to identify what are tumor-specific aberrations. And we are also looking at cancer-relevant germline aberrations in there. So that's pretty standard. The next step is, from those signals that we identify, we are looking at do we have any kind of biomarker-ness of the individual aberrations that we are identifying? So what we need is a good database that contains biomarker information. And that's a challenge in itself. If we look at biomarkers, there are different degrees of biomarkers and different degrees of actually clinical validity. So we have the very well-known endorsed, KOL-endorsed biomarkers. These are the biomarkers that made it to the label, that are endorsed by the ESCO, by the FDA, that are part of NCCN guidelines whatsoever. That's pretty trivial. Then we have the clinically observed biomarkers. And we have different degrees of validity, from really big prospective studies back to retrospective observational studies. And then we have the translational aspects, where we can actually include preclinical information or informatics prediction, so that I call that bioinformatic inference. So if we look at that field, clinically observed, most of the progress that we're having in the biomedical field still is not stored in the form of databases, but publications. So we need to mine publications. We need to extract knowledge from literature, and we need to curate that knowledge. So we have a 4I biomedical expert principle for knowledge curation. So we have a whole text mining, knowledge mining infrastructure that keeps our database up to date. This is just a screenshot of one of our internal tools where you see each line actually represents one text, one abstract or full text section that we have identified, where we relate a drug to an effect, to the context, clinical trial, indication, protein variant, and so that helps actually organizing this kind of knowledge, and we can dive deeper and actually extract that. When we look at clinical trials, we're collecting all the clinical trials that come with biomarker research and a biomarker stratification strategy. So we take these trials, we look into the different treatment arms, of course, also the medications that are being used. And then we are recording for each treatment arm. Here actually the biomarker is a KRAS wild type. We are recording patient demographics, numbers, everything, PFS and OS. So we have these clinical metrics that we can report out later if we have a biomarker match. And actually, the different degree of clinical validity, I always like to match that actually against the clinical need. So and that again depends actually on the individual indication. So for a first-line treatment, I think you would not go for anything that is computationally inferred. But as we have experienced that working together with a phase 1 group at MD Anderson, if you're out of guidelines, if you have a candidate for phase 1 trials, doctors are even ready to rely on computationally inferred information because it's better than shooting into the blue. So something I don't want to spend too much time into, even if we have mutations for which we don't have any reported functional activities or we don't know what that variant actually does, we can use functional assessment. So a lot of heavy bioinformatics, we don't have the time to get into that. So we can generate hypotheses around novel mutations here. That's more a translational aspect of what you're doing. So we need to present this data in a very comprehensive way actually to the oncologist. And what we have created is a web interface that is really summarizing the key findings. And here we're actually looking at a breast cancer patient. We don't need to care about the specifics here. But we have information like evidence and validity for the biomarker, then the drug that is actually recommended, is that available on the market? What's the highest approval status in the indication? Are there any recruiting clinical trials? Does the drug actually cause any negative drug-drug interactions with another drug that the patient is already getting? What is the variant associated? And everything is always connected with the underlying evidence. So we can, actually the physician can mine all the clinical trials information, literature evidence. If we have the clinical background of a patient, he can explore that as well in that environment. And even look at epistatic relationships between genes and mutations and do pathway exploration in that context. So another important thing I already talked about is actually drug safety. And here we have a drug safety profile that we generated for one of the patients. So this is a drug regimen. All these drugs are co-administered with the patient at the same time. And I just want to highlight the patient is getting erlotinib, gefitinib, and clarithromycin, and there are known drug-drug interactions between those. So the risk that this patient is actually running into with a very cheap antibiotic treatment, you can actually kill the effect of very expensive anti-cancer treatments. We map all these drugs actually to cytochromes, transporters, and so on, and we can easily identify cytochrome overload, potential cytochrome overload, especially if you're trying new combinations. That's something extremely important. So what we generate in the end, the oncologist is reviewing this data. It's a trained oncologist, and he can then actually again have his professional judgment, and he creates out of the portal, he creates this kind of a report. This is a medical report that then goes out to the treating physician, and that's actually a real example of a pancreatic cancer case. I just want to focus here on this one. So this is actually a variant that was identified, a germline variant in this patient, homozygous on both variants. And that's an FDA-approved safety biomarker, so there's an FDA warning associated with that. So capecitabine leads to lethal side effects. And actually, if you know pancreatic cancer, metastatic pancreatic cancer, capecitabine and gemcitabine are very yeah, it's not uncommon to give that. So with that combination, you would kill the patient. So it's extremely important also to consider safety. Since we are here talking about colorectal cancer, I wanted to get to one very interesting case that we did a while ago. So here, just for ease of explanation, I have more a pathway, classical pathway-centric view, and everything that we see in green here are actually germline variants in that patient that have an implication on colorectal cancer. And all the red ones are somatic mutations in the cancer. So we see the classical APC nonsense mutation. We have a MYC, gain of function mutation, actually a phosphorylation site that triggers degradation is hit by the mutation. So it has a longer half-life time in the body. But I want to focus especially on what we increasingly find the more we look at cancer patients is we have a predisposition, actually a germline biomarker in one of the genes, and we see somatic hits in a functional vicinity to that one biomarker, to that one germline variant. In this case, the germline variant is in PI3K receptor, and it has been described in literature that actually this has an increased affinity to IRS1, although it has no impact on cell growth by itself. But when it's in combination here with an upstream mutation, so IGF-1R is recruiting IRS, is recruiting the PI3K receptor, is recruiting PI3K, then it actually has an amplifying effect. And here we don't have only one mutation in the vicinity of a germline biomarker. We have another somatic mutation here in PI3K that is of clinical importance with actually a lot of data. So just in very brief, what we did with that patient case. So we have a KRAS mutation here. We have PI3K activation. We have some activation of RTKs. So we have activation, and we have seen this, these pathways before from the colleague from Bayer, for example. We have an activation of RAS-RAF-MEK-ERK, another activation line actually along PI3K ACT-mTOR. And what we have listed here is now for these pathways all the different inhibitors that are in clinical development at various stages in various indications, most of them early stage, so all kinds of solid tumors. So actually what we do in such a case is we filter for clinical trials that are actively recruiting patients and that are in geographic vicinity to the patient. And these were the two trials, those days that case was done 1 year ago, that were open for recruitment that are actually combining PI3K inhibitors with MEK inhibitors, one by Bayer and one by Novartis. So that's one of the classical examples. So in the end, my firm belief is cancer genome sequencing is not going to give us the truth. It's going to give us a very important building block. It always needs to be seen in the context with other diagnostic tools, other diagnostic biomarkers. But since we're really looking at the genetic cause, at the cause of the disease, I believe it's a very important and very informative building block for the future diagnostics in cancer. Thank you very much.
Thank you, Stephan, for this excellent presentation. Mind-boggling graphics, too. Questions? If I may have one now, the way you work with literature and the way you work with sequences, that is not necessarily, the input information is not necessarily restricted to just sequence information if you work with the literature as you do.
Yep.
Can you comment on that?
Absolutely. So I mean, the focus on sequencing is really, it's commercially driven. I mean, we want to get to a kind of a standardized test. You can, of course, analyze all kinds of information. I mean, we've had patient cases with collaborators at big cancer centers where we had RNA sequencing, we had array-CGH, we had methylome data, we had differential expression, and we had full genome sequencing. And we analyzed all of that. But when we think about offering a straightforward standard test, I mean, one of the limitations we have is the original tumor material. And if we think about clinical practice out there, FFPE is the standard, and FFPE is not suitable really for differential expression for RNA analysis. There are now some kits, but then you have to follow exact procedure on how to treat the tumor material when you prepare your FFPE. So it's very practical considerations, and it's also a matter of price point. I mean, it's still expensive, but if we add more technology, it's even more expensive.
Okay, thanks again, Stephan.